Um, it gives me great pleasure to welcome a friend, and actually not only a friend, but he was my chief at Walter Reed when I got there. He was the chief of neurosurgery, the chair of neurosurgery. Kevin, let me tell you what he is now. He's a professor in the Department of Neurosurgery at the University of Tennessee. But more importantly, they have kind of a bifurcated leadership. He runs Semmes Murphey Clinic, which has been in practice over 100 years, Kevin, over 100 years. So he is the chairman of the board. I like that. That's better than your title. He's chairman of the board of Semmes Murphey Clinic. To tell you my history, fascinating background. Kevin is an Army brat. Kevin grew up on Army bases throughout the world. His dad was an armor officer. Those are people that drive tanks. Okay. His last station, his dad, may he rest in peace, was as commander of Fort Knox, which is a big tank school that they teach people how to drive those things and fire them. He went to Deerfield Academy. He's like Jeff Ogeman, brilliant that he is. He graduated. He finished his college in 1 year. I had a hard time finishing it in 4 years, but be that as it may, he went on to UCLA, where he graduated with honors in AOA at UCLA, and more importantly, met his very successful and wonderful wife, Lynn, who they've been together for over 41 years, and they have 3 children. Not surprising that they're all brilliantly successful, but none of them went into medicine, right? Exactly. Smart children, right? What was Kevin's superpower? I'll tell you my perspective. Kevin, if you look at his CV, he has a lot of publications, obviously, from all the work he's done in spine, but he has more patents. And they're all patents. You look at, he says, wait, he did that? Well, what he did when he was at Walter Reed, he had all these great ideas. I mean, imagine this was 35 years ago. And he said, surgery is going to become minimally invasive for the spine. And it was just getting maximally invasive. Scoliosis surgery was big time surgery. And he says, someday they're going to be putting in screws percutaneously using imaging. And everybody laughed at him and said, that's the craziest thing. So what Kevin did is he went out and invented the systems that we now use today. So he'll tell you that he's helped more people with his inventions and innovations than he's helped. He's still a busy surgeon. I mean, obviously after being in practice at Semmes Murphey for 35 years, people come from all over the country to have him do their surgery and go home a few hours afterwards. So the other thing, Kevin's just a wonderful human being. I tell the story when I got deployed to the Middle East during the conflict. The deal was that my wife was in the Army. She was active duty as well. And the deal was that it was not uncommon to deploy the wives and the husbands at the same time. And they said, just leave your kids with the next door neighbor. The problem was we didn't know our next door neighbors, and my wife was pregnant. And so, and she was the most deployable and useful because she was an ICU nurse. She was actually useful in a wartime scenario. And back then, pregnancy leave was 3 weeks. The Army gave you 3 weeks, and then they sent you. So she was getting deployed 3 weeks after my son was born. My second child was born, and she got 3 weeks, and they were deploying her. And Kevin, the genius that he is, figured out a way to MRI all kinds of parts of my wife's body to find something abnormal. And he never did, but somehow the bevy of orders for MRI kept her from being deployed during wartime with a newborn baby. So Kevin, our family owes you a debt of gratitude forever for doing that. But I don't want to take up too much time, but Kevin obviously has done this better than anybody else in neurosurgery. And the secret about Kevin was that he understood, and it ruffled feathers. And I said to him, where are you going to go when you finish at Walter Reed? You know, we had a wonderful time at Walter Reed. It was a very inventive time. The neurosurgeons that were in the Army at that time, Kevin Foley, Reggie Haid, Chris Shaffrey, really invented modern spine surgery as we know it. And it was all because they were inventing things, new devices. They were taking things from Europe and perfecting them. And so they shaped a field, and Kevin said, I'm going to keep my IP, which was ridiculous idea. You couldn't keep your IP. If you worked for a hospital or university, they took your IP. Kevin says, I'm not joining any institution that takes my ideas and then use it and then takes the profit from that. And so he figured out a way to do this legally and morally. And as a result of that, Kevin has been richly successful. Kevin didn't have to work for 20 years, but he still works and still loves neurosurgery because he grew up as a first-generation physician, and he can't believe someone lets him do this for a living and pays him to do it. Without further ado, I want to introduce a great friend of mine and a brilliant innovator and the guy who does it best in our field in terms of invention and entrepreneurism. Kevin, thank you.
Well, that was an overly generous introduction from your chair. You know, it's my pleasure to be here this morning, and you know, have been friends with your chair for over 30 years, back to when we were young guys at Walter Reed, and I'm sure you all are aware of how fortunate you are to have such a good person to work for in running your department. So topic here is self-evident. I'm going to talk to you about innovation and entrepreneurship in neurosurgery. These are my disclosures. We do this whenever we give academic talks, right? We want to talk about anything that might be a conflict of interest, important to disclose so people can understand that there may be biases, there may be hidden motives, etc. But these disclosures for this morning's talk are very, very pertinent because these all relate to things I do in my work to develop new ways to treat patients. So my day job, my vocation, you know, we're busy creatures as neurosurgeons, and it's as you know, the neurosurgery is a demanding task and quite time consuming. And it becomes a passion for all of us. I don't think we would do it otherwise. But I'm still a busy surgeon. I operate 3.5 days a week. I see patients a day and a half a week. I run a large neurosurgery clinic. We're also the academic department at the university, so I'm a tenured professor there. And the box on the right is from Google Scholar. You know, I've got a reasonable H index. I've been cited more than 55,000 times in the academic literature. So, you know, I've done okay, and that should be enough, right? But no, because my hobby, which is really my other passion, is inventing things and trying to advance the field in that fashion. This is a generative graphic. You know, you can go to DALL-E and some of the other generative graphics websites. This is from Adobe's. It's called Firefly. But the query, if you go to Firefly, they say, "What do you want this picture to show?" And so I said, "Neurosurgeon inventing a new surgical product," and this is what the computer spit out. So I do this, right? Again, we're busy enough, it's hard enough, time consuming enough, demanding enough to do neurosurgery alone. Well, primarily, it lets us solve problems, and we all solve problems as neurosurgeons every day. And And those solutions sometimes require a new pathway, a new way to do things. And you can absolutely help patients this way, as Rich said. You know, I know that I've touched millions of lives with things I've invented, and I've touched you know, tens of thousands of lives as a surgeon. But you know, surgery as great an endeavor as it is, it's piecework, right? We're like seamstresses, and you can only sew so much. But if you come up with a great solution that your peers adopt, you can touch many more lives and help advance the field. I also believe we're in the best position to do this. Though there, as you know, there are companies out there and other entities that help provide solutions for medical and surgical problems. But because we're on the front lines, because we see these problems firsthand, we are in the best position to advance the field. It's absolutely enjoyable and intellectually stimulating. And finally, it can allow for a financial cushion to allow you to practice medicine in the face of, you know, constantly diminishing reimbursement. So that's the least important reason to do this. It's a pleasant byproduct of this. So many people aren't aware of what's happened over the years. We all know that physicians are not getting the resources that they got 20 and 30 years ago. And you'll read, well, reimbursement's down 20%. Well, it's down a lot more than that. So this is from one of the government's inflation calculators. And I picked 1984 because I finished my training in 1985 at UCLA. So if you had something that cost $1 in 1984, today in 2024, it's $3, that same dollar. So let's translate that to some common procedures. So a laparoscopic cholecystectomy. General surgeons were paid $2,000 to do that in the 1980s. Okay, that's $6,000 today in 2024 dollars. You know, general surgeons now get between $300 and $400 in 2024 dollars to do that procedure. Unbelievable. 3-vessel coronary artery bypass, $4,000 in the 1980s. That's $12,000 in today's dollars. They get $1,800 to do this. This is in our area in the Southeast. It's probably similar here. In fact, one of my friends, great cardiothoracic surgeon, he just joined a hospital practice. He was the last private practice cardiothoracic surgeon in Tennessee, Mississippi, Alabama, and Louisiana. They've all had to join hospitals because they can't afford to run their own offices with the low reimbursements out there. In our field, and there are numerous other examples, but a common procedure, a lumbar discectomy, $3,000 in the 1980s, right? We get a fraction of that today. And so that trend is going to continue through your careers. Get used to it, because we're still relatively well paid compared to physicians in other countries, and we're a soft target, as they say in the military. We're very easy to attack in this fashion. And this is not to dissuade you from going out and taking very good care of patients. That's what we do, and we're still, you know, nicely rewarded for that activity, but it is getting harder and harder to run an independent practice. So how would I do this? Rich mentioned this. We were talking about this at dinner last night. He knows that when I was at Walter Reed, I was already dreaming up some things that I thought we could do better. The first thing I ever invented was at Walter Reed. And lo and behold, found out that, wait a minute, I'm a government employee. That's not really my invention. I had to cede this to the US government. And so I'm a rule follower, followed the rules, but I also learned. When I finished up at Walter Reed, I had the privilege of being the chairman of neurosurgery there, was getting some nice academic recruitment offers all over the country. One of the things I resolved, Rich mentioned this earlier, was said, you know, I'm a neurosurgeon. I'm coming to do neurosurgery for you. You've got me there. That's why I'm here. I'm not here to invent for you. That you're not hiring me as an inventor. You're hiring me as a neurosurgeon. And so my inventions that I do on my own time without using your facilities or your resources are mine. And that was one of the reasons I ended up at Sims Murphy Clinic, because the Sims Murphy board got together at the time. They also wanted to own your intellectual property. The board got together. They wanted me badly enough. They resolved that, well, if you do it on your own time and with your own resources, then yes, that IP can be yours. And that rule made, 33 years ago when I joined Sims Murphy is still our rule at the clinic. Again, it has to be on your own time and it has to be using your own resources. So one of the first things I came up with, solving a problem, was a way to get a lot of screws to line up. Now, a pretty simple problem, but there wasn't a good answer at the time. Posterior cervical surgery at that time was done with plates and screws, and the plates had linear arrays of holes. And so if you were doing 1 or 2 or 3 levels, relatively easy to line up those screws. If you were doing 7 or 8 levels, you're going C2 to T2, it was a difficult task. So I came up with a polyaxial screw. They're common today, right? They didn't exist then. And I partnered with a little company that was in a storefront in Memphis at that time called Danik. And they assigned an engineer to the project. We came up with a very nice system. And that working relationship formed. But, and I'll talk about this a bit more, the downside was they were already selling a plate, which was a knockoff of something called the Roy Camille plate. They called it the Axis plate. And so they really weren't interested in productizing this. And so my IP got shelved for a decade. It's one of the disadvantages of working with someone who ends up controlling what happens with your ideas, not necessarily the ownership thereof. So along those same lines, at an academic meeting later that same year, something called the Society of University Neurosurgeons. I don't know if anyone, Rich, belongs to the Sun Society here. I met Rich Buchholz, who was a young neurosurgeon at St. Louis University at the time. Went up to his attic lab at SLU, and he was working with a young engineer, Kurt Smith. And this was the beginning of what is now called the stealth station. They had, a camera array, a very primitive camera array, a digitizer, a computer for graphics display, and basically had a very rudimentary but functional system to navigate in the head in a frameless fashion. Mind you, all stereotaxis for almost a century before that and up to that time was framed. No one was doing frameless stereotaxy. But also, Rich and Kurt weren't doing anything in spine. And I looked at their system. I said, guys, I can make this work in the spine. And they said, okay. Well, they were also looking for some funding. And so I agreed to join their effort as a co-founder. Took one of the computer systems back to my lab in Memphis, and basically developed a working spine prototype. Well, fast forward a couple of years, and like happens with many, many startups, we were running out of money at Stealth. And so Rich and Kurt went out and about to try to find companies to buy us or invest in us. And interesting, all the major players at that time, dominating framed cranial stereotaxis, were not interested, not at all. So Rich and Kurt were a bit crestfallen, called me up and said, Kevin, I think we're going to have to end this. I said, no, let me call a friend. And the fellow I knew through my work at Danik was Ron Pickard, who was their CEO. Now, Danik had nothing to do with cranial surgery at this time, zero. But I knew that Ron Pickard was an innovative thinker, and I invited him over to my lab in Memphis. I said, Ronnie, I want you to come see what I'm doing. I think this is going to be something that is going to be useful for people. and on a cadaver, navigated. And on the spot, Pickard said, I want to own this. I want to buy this. And that's how Danik got Stealth. And that's why Medtronic has Stealth today. They bought Danik. But an example of collaborating, working to solve a problem, coming up with something that literally has helped change the world and I think definitely improve patient care. This, by the way, this is from a book called Intraoperative Neuroprotection. I published some of our early work back in the 90s. That top picture is that's the first spinal dynamic reference array. So I patented dynamically referencing the spine. No one had done it. The field was wide open. And I went to the hardware store, as I would often do, looking for solutions, and thought, a vice grip. I can clamp anything with that. And so I attached LEDs to the vice grip, and that was the world's first spinal dynamic reference array. And a drill guide, I took a Danik Axis drill guide, that same plate system that I talked about earlier. I said, I'll put some LEDs on this, and then I can look at that trajectory on the computer screen. Anyway, that was the beginnings of Stealth Spine. So, kind of an overview of what I've done over the years. I have a number of patents. I've invented several things: microtubular surgery, spinal navigation with dynamic referencing, percutaneous screw rod fixation, the minimally invasive TLIF, virtual fluoroscopy, the notion of tapering the nose of interbody implants. No one had done that before, and a lot of other things among those 182 patents. And I've founded or co-founded through this journey a number of companies. So I was a founding partner of Stealth, which we sold to Danik, as I mentioned before. Co-founder of a company called Spinal Graft Technologies. Was a partner and board member of True Vision. We built the first digital microscope for ophthalmology, and spun off from that another company that still exists down in Santa Barbara, True Digital Surgery. Was a member, a founder and a partner at BioD, which we sold eventually to Integra. Triad Life Sciences, focusing on tissue repair with porcine tissue. We sold for a very nice sum to a large British American company, and then a number of other things that are listed here, and I'll talk about some specifics. But doing this honestly part-time, doing this on nights and weekends when I'm not being a neurosurgeon. So startups sound very, very intriguing. Everyone thinks, oh, I'm going to do very well with my startups. There are challenges, lots of challenges. Remember that 90% of startups, I have it underlined at the bottom of this slide, 90% of startups fail. And the primary reason that they fail is they run out of money. It's very, very difficult to go from startup to existing company or exit. Other challenges, things that were never taught in neurosurgery. There are market issues, there are product development issues, not just innovating, but then manufacturing and quality control and packaging and sterilization. Again, things we don't typically think about. You have to work with a lot of other humans, not just running a department of neurosurgery, but running various divisions in your company and managing a team and leading that team. Legal and regulatory compliance. Regulatory compliance alone can shut you down. Navigating the FDA is an entire and complex endeavor. Scaling the effort, managing your risk, and then how do you exit? Do you ever exit? Are you planning on being acquired? Are you going to go for an IPO? Are you going to merge with another entity? Complex things, again, that we learn nothing about in neurosurgery. I've learned over the years. Some of these things I've learned the hard way, and I know you've got some fellow faculty who have been down this road. So another way early in your careers to innovate is to collaborate with an industry partner. It's a much easier way of bringing your innovation to market than a startup. And it is gratifying to see something that you developed or helped develop out in the field being used by your peers. But this is time consuming, but it's a lot less time consuming than going through the startup pathway. And so a lot less capital intensive. Trade-offs, I mentioned loss of control. You know, I invented polyaxial screws, but there were no polyaxial screws on the planet. And at that time, could have built a huge industry right then, but no, the company I was working with shelved the idea for a decade. And although most startups go out of money and you can lose a lot of money pursuing a startup, a successful startup will typically yield more financial gain than you'll get with the collaboration pathway I'm talking about. But still, that's a relative a downside. Very, very importantly, and this is important in life, it's important in medicine, it's very important in business. These things allow you to build relationships. As people learn to trust people who are productive for them and turn out to be honest and hardworking, just like you know, think about how what you like in a junior resident. You know, you're going to teach them, but you want someone who's absolutely honest, absolutely shows up on time, absolutely does their work. It's true in all aspects of life. So the collaboration pathway allowed me to create some of the things I talked about a moment ago. In all of these things, my motivation was to solve what I perceived to be a clinical issue, trying to make care better. So when I was a young surgeon, I'm sorry, Christoph Hofstadter's not here. I know he's out and about, but endoscopic surgery had been around since the 70s. Hijikata in Japan was the first guy to do it, but it didn't work well. And I was intrigued. As Rich said, one of the things I wanted to do was make surgery less invasive. Lumbar discectomy back in the day, people had a 6-inch incision and stayed in the hospital for several days after a lumbar disc, right? Now you'd say, well, unbelievable, but that's just how it was done. So endoscopy was fascinating as practiced then because it was so less invasive. Unfortunately, it wasn't routinely effective. So you not only have to be minimally invasive, you've got to be maximally effective. So I worked in my lab. The lab, by the way, is a private lab I set up. So that's not my university lab. That's my lab off campus. Worked on basically shrinking the approach. But what I wanted to do was get 2 hands in there, and I didn't want to hold the endoscope because I wasted one of my hands. And so pretty easy things to think about, right? Let's be able to get 2 instruments in so you can work with both hands as we do as surgeons. Why work with just one hand? And let me not waste one hand holding the scope. So that evolved into what became MED, microendoscopic discectomy. At the time, I was doing it endoscopically because the focus was endoscopic surgery. But I would park the endoscope in the tube. I would attach the tube to the table so that I could maneuver it, but then lock it in place. And then I could use both of my hands like we do in standard neurosurgery. And it turned out that worked. Started in the lab. We did the first patients in the mid-90s and found out that, gee, I can do more than just decompress this thing. I can put things in through this little tubular approach, of course. Couldn't do good internal fixation, which led to another problem that needed a solution. But this is the publication of the initial case series of MED. You know, it's interesting, nothing new under the sun, as King Solomon said. So people say, oh, we're doing awake surgery. So all these cases, this was published in 97, but the case series started in 95, all outpatient, all awake, under local or epidural anesthesia. So, you know, nothing new under the sun, and doing not just discs, but stenosis and the like. And this worked. It turned out, though, that you could also look down the tube with a microscope. And unfortunately, one of the downsides of endoscopy is it's costly. If you look at the cost of goods to an OR for doing an endoscopic case, and we already have these microscopes sitting there. And lo and behold, if you could do it with a microscope, you could do the same thing now in 3 dimensions quite easily, and a lot less expensively. So continuing to solve problems, we developed the minimally invasive TLIF concept. Initially did fusions without internal fixation. This is in the early days, gang, of interbody fusion. Ralph Cloward, one of our forefathers, had developed the PLIF in Hawaii in the 1940s, working on laborers and seeking a solution. It was quite controversial at the time, but it became the foundation of posterior lumbar interbody fusion. None of those patients were instrumented. So now we instrument them all for various and sundry reasons: deformity correction, better fusion, et cetera. Well, there was no way to do this optimally as I approached this problem. In fact, this was the way. Okay. This is called the fixator extern. It was developed by Fritz Marble, surgeon in Europe. And the fixator extern came from general orthopedic trauma, because you could put these percutaneous screws in and fix other fractures like pelvic fractures. But you connected them outside the body. So like that giant erector set sat outside your back, these long screws called shank screws would be targeted down the pedicles and then connected outside. Patients had to wear this bubble brace, because you could imagine if you leaned back in a chair or in your bed, you'd snag all of this stuff. Infection rates were not trivial, and you had to take this out, right? This hardware required yet another trip to the OR. So I thought, surely there's a better way to do this, right? There just didn't exist a way at the time. Well, it led to the development of this. It occurred to me, and this was because I was doing image-guided placement of implants at the time. You see it all the time now, but I could digitize an implant, put a CAD of that on the screen, put that into first models and then cadavers and then patients, and see, oh, there's where that thing is. Even though you couldn't see it visually, you could see it on the screen. And then I thought, well, boy, if I knew where these screw heads were in space, and especially how they were oriented, and then I could detach all of this stuff, I bet I could fish a rod down there. And that turned out to be a really good concept. It underlies what you guys probably do every week here. Okay? And it's done all around the world now. But the genesis was solving a problem. By the way, co-author here, Sanjay Gupta, known much more now as medical correspondent. Sanjay was my fellow at the time when I was developing this, and got the call to become the CNN. They wanted him to go interview to be the CNN medical correspondent. I was talking to him one day. He said, "What should I do, Dr. Foley?" And he was talking to his dad. He was talking to Buzz Hoff, who was the chairman at Michigan. Sanjay, what do you want to do with your life? And he said, I want to be a U.S. senator. I said, well, if I wanted to be a senator, I said, neurosurgery is a great thing, but we're big fish in a small pond, right? People don't know us. I said, you want name recognition. If you're the CNN medical correspondent, you'll have much more name recognition. Anyway, interesting but true story. Okay, so stuff I'm up to now, and as Rich said, I've been fortunate enough. I've done very well with some of these inventions, enough to, a lot of people would retire. I love being a neurosurgeon. I love taking care of patients. That's why I still operate. But I also like still solving problems. So it's what I do. I'm not a golfer. I don't play bridge. I do this. So working on disc regeneration, I'll talk about that. Developed, I was looking at these $1 million pointing machines, the current generation of spinal robots, thinking, oh my God, there's got to be a better way. came up with a very low cost, easy to set up robot that we ended up selling to a company called ATEC last year. AI buzzword. Sam somewhere in the audience, I'm sure. I'll show you one application of artificial intelligence. Some interesting bone glue stuff. Some different ways to image in a low dose fashion, but very fast registration. All of these efforts are things that, in my mind and in my experience, aren't done as well now as they could be. Doesn't mean these are all going to be the answer. Maybe some of them will, but I'm pursuing them because I think they're a better way to try to deal with some of these clinical problems. So dysgenics. This was research that started at UT, University of Tennessee. UT decided that it was done within our department. Now, this was done on our university side. And so at our university lab, the university decided as a science project, they didn't want to continue to pursue the IP. They didn't want to spend the money. And so we approached them, a couple of us, and said, will you license that IP to us? And they said, yeah, absolutely. We're not going to pursue this. So fast forward a decade, and this is now a company in Salt Lake City. And we just finished our phase 1, phase 2 FDA trial, and about to start enrolling in our phase 3 clinical trial in December. But the disease we're trying to treat is chronic back pain from degenerative disc disease. And not to get into it in great detail, but it is the most common cause of chronic low back pain, most common cause of lost work days, not only in the US, but throughout the world. And, we don't have a biologic solution to this disease. We've got, when people fail all forms of non-operative management, are you going to put an artificial disc in there? Are you going to go in there and ablate their basovertebral nerve? Or are you going to go and fuse them? None of those things is ideal. So we have developed a cell population that can regenerate these discs. That's the reference down at the bottom. Just published that in the International Journal of Spine Surgery in June of this year, the results of our phase 1/2 trial. So, which is another nice benefit of this activity as an academic neurosurgeon. You can get some academic productivity from this as well, from your publications. So that's a discogenic cell cluster in the spinning graphic on the, that's actually from a confocal microscopy picture. But these cells, we get them from beating heart donors, and then we drive them backwards in the lab. The culture technique is loosely based on the neurosphere culture technique for deriving stem cells from the adult human brain. Some little variations on the theme there. Bottom line is they become progenitor cells. They lose their immunogenic cell surface markers, so they're non-immunogenic. In fact, in all our animal studies, we used human cells, never saw any rejection. They have anti-inflammatory properties, but they regenerate too. They begin to immediately, even in culture, they begin to make the constituents of a normal disc. And so in the disc, it's a tough environment. These are the disc is anoxic, the disc is hypoxic, the disc is acidic, right? So most cells, like people say, I'm going to take a mesenchymal stem cell and put it in the disc. It's going to die. They are not. Discs are, it's an unfriendly environment. That led to our thesis of saying, well, geez, let's take cells that already are designed to live in the disc and see if we can't make them work. So that was the genesis of this work. So this is, I won't bore you with the trial. Basically, patients had to have chronic mechanical back pain that had failed a good trial of non-operative management, and it didn't have instability, didn't have neural compression. So these are back pain patients who had failed non-operative management, and they were randomized to a low dose of cells, a high dose of cells, the vehicle alone, which is hyaluronic acid, or placebo, normal saline. In cell-based research, I'm sure much is done here. You move from the device world into the drug world. And so this happens through the drug branch of the FDA in an investigational new drug application. Very, very, very rigorous requirements, as you might imagine, before you're allowed to use this stuff in humans. So years of animal work, preclinical work preceded this. Subjects were blinded to treatment, basically got an injection of either the low dose, the high dose, the placebo, or the vehicle, and then followed. And I'll show you the graphics, easier to see. Interestingly, saline has an effect, just diluting the disc with some saline, and perhaps you're even temporarily hydrating it. You notice if you look at the pain score here, black is saline, the blue line is the high dose cells, the orange red line is the vehicle, and the green line are the low dose cells. So out at 2 years, we reached statistical significance with high dose cells, low dose cells, and vehicle. Saline, actually early on, we thought, what do you know? We were seeing a response, but that tails off, and by 2 years, that was non-significant. The point of the slide here is that with the high dose cells, we saw the earliest response, and then that diminished pain was consistently maintained out to 2 years. More important was we also looked at the patients functionally. So this is looking at their Oswestry Disability Index. Significant improvement in function in these patients with the high dose cells, over twice what's called the minimum clinically important difference. Same thing in their quality of life is measured by the EQ5D. High dose cells are at the top. Higher EQ5D is better quality of life, almost 3 times the MCID. And then these are the combined graphs of the outcomes of the high dose cells: improved quality of life, diminished pain, and diminished disability. But we also did imaging of these patients. The patients underwent MRI, and the MRIs were all read by a core lab in Medical Metrics, which is in Houston, with an automated algorithm that can measure volume on MRI. And here's the volume data. So we had seen this in the animals. We followed the disc height index, and we could see discs that would diminish in height with experimental intervention. There are experimental models of disc degeneration. We see that height restored. And here you look at the high dose cells up top from baseline. 0 is the baseline. Continuing to improve in disc volume even at 2 years out. We were excited by that, and FDA was too, because it was some evidence that there was a regenerative effect going on in these discs. So phase 3 trial, where we're recruiting centers. Rich, I don't know if you guys are interested, but it might want to join the trial. AI, an example of that. So TDI. What's the problem we're trying to solve here? The problem is doing transpsoas surgery and not knowing where the nerves are. People still clobber the nerves from time to time, especially at L4-5, right? And it's because evoked EMG isn't perfect. You can't find any sensory nerves that way, and you can miss some of the motor nerves. So one of the things that generative AI algorithms are good at is processing of digital images, right? If you look at the applications of AI in medicine right now, radiology applications are number 1. I don't know if you guys are using Viz AI, for example, in your endovascular work. We use it routinely. And cardiology number 2 right now. Okay, we're coming along in neurosurgery. So here's an early neurosurgical application of that. We took a beam mode ultrasound image and trained it with a convolutional neural network to find nerves. So Tyler Carson, this publication is the publication that resulted. This is in Spine for reference. Tyler, my fellow that year, I stuck him in, he spent all his nights in the lab, because you have to train these algorithms. And we said, we're going to do this in cadavers. Well, it didn't work in cadavers. The tissue characteristics of non-living tissue were different. So we did the early development work in pigs, and have to anesthetize the pigs. And Tyler would target these nerves ultrasonographically using the algorithm, and then have to have a ground truth. And so we'd follow a stainless steel pin that we could see ultrasonographically target the nerve, and then you have to dissect down to validate whether the algorithm was right or not. Through a year of working through that, came up with a really effective algorithm. A good example, that image to the right that you see here, that's the B mode ultrasound. So where are the nerves there? What is a nerve? It's hard to know. The human eye can't pick that out as well. And so with the TDI ultrasound probe, you put it in, you get your B mode images, you park it on the psoas, then you flip the switch and things show up. That yellow thing is a nerve where you don't want to go. Those 3 channels, we have 3 holes that you can use for your access into the disc. And obviously you don't want to use one of the access portals that might transect a nerve or injure a nerve. But the B mode can be trained with this algorithm to find the psoas, to find the contours of the bone, that you can see the cortical boundary there in black. And there's a large nerve at L4-5 sitting right on the vertebral body. That's from a human case. So an example of problem solving. I will tell you, we have not proven yet that this is better than electrical testing, but I think it's going to replace electrical testing. And we're using it in some other interesting ways too. There are some other places where it's kind of nice to be able to see the nerves down deep. This is another effort, a small company out of Boston that I'm the chief medical officer for. So for decades, going back well before I became a medical student, we've used bone cement, right? It's inert. It doesn't adhere to anything. It's hyperthermic when it's setting, and it's a foreign body. If you get an infection with the stuff, it's a mess. But we don't really have anything else. So the picture on the left is those are sandcastle worms. And the sandcastle worm, it's a sea creature, a marine creature that builds. It's not really a sandcastle, but it builds its house, so to speak. And the substance that it uses is something that our group has taken and developed a bone adhesive that can turn into bone. The applications here are enormous. In business, they talk about TAM, total addressable market, right? There's lots of things you could do with an effective bone adhesive. And I'll show you some of the experimental work. So plates and screws in the head, right? That's what we do. Years ago, we used sutures, or we used wire. Rich remembers this, right? Then plates and screws. Oh, wow, this is great. But, it turns out that the kerfs, the gap that you cut with your craniotome between the bone flap and the skull, hardly ever heals. Right? You get lots and lots of fibrous union, and that's why, we have some issues. Flaps that migrate a little bit, hardware that they're out a year, and they're saying, "Yeah, doctor, it bothers me here," and you're thinking, "Be happy you're alive from your brain surgery," but they really are focused on this bump in their head, and they've got a little loose screw there. And the reason they do is that flap is toggling. It's not actually got a solid union. So this is from a lab study I published in Journal of Neurosurgery a couple of years ago, from a sheep study with our bone cement. So these are sheep craniotomies, and each sheep was its own control. So biparietal craniotomies using a Treffen. So it's a 2 centimeter cranial bone flap on one side, plated, and on the other side, glued with the bone glue. And did biomechanics, won't go into all the details, but the histology is interesting here. So there's plate fixation in that middle row, and bone glue fixation at the bottom. So the blue on this staining is collagen, and you notice that even at 2 years, lots and lots and lots of collagen in these kerfs. And so the union rate, and this is true in humans as well, is not ideal when we're using plates and screws on these patients. And contrast that with the bone glue. And what this particular bone glue does, it's osteoconductive, it's actually osteopromotive as well, but slowly but surely bone grows through it. So you can see by 2 years, you don't see nearly as much of the adhesive. You see living bone growing right through this. So we did another study. So look, great, this is great and cheap. This is before we started our human trial, looking at fixing bone flaps in human skulls and just testing these things biomechanically. So a variety of craniotomy flaps, randomized to half with plates and screws, half with bone glue, and done here at the Brigham and at Geisinger. And we recruited our partners to be they would get to glue one flap and then they would get to plate the other flap. So one of the things this not only osteoconducts, it adheres and it seals. And so hydrostatically, we showed that, of course, with a kerf with plates and screws, saline would pour right through. But we could pressurize the flap up to 40 millimeters of mercury and see no leak. So in cases where you want to try to preclude CSF leak, maybe there's an advantage to that. And then looked at the force that these flaps would sustain shortly after fixation. So we fixed them, and then we had this setup that I'll show you right in the lab. This was static testing here, but here's the dynamic testing, which is kind of interesting. So this is we can accelerate this weight on the flap. Look at the plated flap with 6 joules of energy on the left, and there's the same flap location and size at 6 joules and even at 12 joules. And if you look closely, those other the flaps they're glued don't move. The stuff is very, very strong. In fact, it turns out it's as strong as the intact skull right after you glue it, right after it sets. And so, what are the implications of that? Well, think about returning an athlete or a child to play after a craniotomy. The impact energy of a soccer ball moving at 10 meters a second is 22.5 joules. Well, I just showed you what happens at 6 joules with a plated spine. You wouldn't want that kid playing soccer or baseball or any contact sport, probably for at least a year after surgery, maybe longer. I think with an adhesive, you could send them out relatively, much earlier. And those were some of our conclusions. So another publication from that, also in Journal of Neurosurgery, but this was predicate work to get us into an FDA trial, which we're just beginning. And FDA, our early cases had to be cases where there was no dural opening. We did animal experiments to show the FDA that even when this stuff gets on the brain, nothing happens. It's interesting. I went to the literature to try to find, we said to the FDA, we've done craniotomies for years, when we do craniotomies, how sometimes the craniotome will catch the dura. We've all done this, and you've now got a dural opening along with your cranial flap opening. But by and large, we don't believe craniotomies damage the cortex, right? We'll go out and try to prove that, right? And so FDA looked at our histology from some of our animal studies and said, look, we see a little tissue damage on some of these cases. We think it's your glue. We said, oh, that's just what happens during craniotomy. Well, they wouldn't buy that. So we had to do, and we'll end up publishing this. We said, oh, surely someone has looked at this in the literature. No one's ever looked at it. It's one of those things we just take for granted as neurosurgeons. So anyway, we did a study on that to show FDA that no, this has nothing to do with the glue. This is all mechanical trauma from cutting the bone flaps. So based on that, they allowed us to start the human clinical trial. This is actually one of the first cases. It's a patient who had a big bone flap for one of the wonderful things we deal with all the time, which is huge brain swelling after a stroke. I'm sure you guys do lots of decompressive craniectomies, right? And eventually, if they live, they come back. They want the bone flap back. So this is the day after the flap was placed, the CT and the plain film. Sorry for the head positioner there on the CT scanner, but you can't even see where the craniotomy was. Pretty interesting. We've just finished the first 6 cases. We've got to follow them for 6 months to show we haven't seen any issues, but FDA wants us to follow them for 6 months. And then we're going to start a general trial in craniotomy. Another thing, Rich, that I'd invite you guys, if you're interested, we're going to be looking for sites to do this. You'll randomize patients to either standard care, which is plates, or to the glue, okay, for craniotomy. So lots of examples of things that were really directed towards solving clinical problems. So lessons that I've learned. I've learned that innovation is an extension of basic neurosurgical problem solving. And we routinely solve problems. We deal with clinical problems every day, if not in the clinic, certainly in the OR. And we have to find solutions to these things. So, but there are other problems that we don't have the best answer to. There are always better ways to do things. It's why I think we as neurosurgeons are in the best position to solve these problems for neurosurgery. You have to learn a few things to do this. It is important to learn how to develop, apply for, and protect intellectual property. And you have to also know the ground rules that are applicable to you. Your institution will have rules about your intellectual property. Lots of people never bother to check and sometimes find out the hard way. But you do want to make sure that you protect your contributions and that they're valued in some fashion. And then you have to learn some business principles to navigate this. And we're you know, one of the things I don't like about what's happened in medicine over the past many decades is bit by bit, our forebears basically ignored the business of medicine. You know, we're busy, we're physicians, we're taking care of patients. We let the administrators do things. And I'm sure you've seen the famous graph Rich and I were talking about this morning. Supply of physicians over the decades since the 50s, flat, okay? Supply of hospital administrators, it's like this. And I don't know about your hospital administrators, but I can tell you ours make a lot more than our doctors do, okay? It's like, gee whiz, what's going on here? You want to learn something about business. Trust me, it will serve you well over your career. You want to learn something about especially negotiation strategies and how to value things, okay? People otherwise will take advantage of you. You are going to need attorney help, whether it's in protecting intellectual property or reviewing contracts or structuring businesses or dealing with the taxes, dealing with your institutional rules, et cetera. Some really basic stuff. How many people know what a provisional patent application is? Show of hands. Okay, so a few. So you all should know what a provisional patent application is. We used to be in the US what's called a first to invent nation. We were one of the few. Most of the world is first to file. First to invent means if I invented it first, it's mine, even though you maybe copied me and went and filed a patent before I did. Well, think about it. Big companies, if they employ patent attorneys, they've got reams of helpers. You're by yourself, right? And so they can get something filed at the patent office a lot faster than you can. In about 2013, the America Invents Act was passed to great fanfare. Oh, this is for the little guy. Complete rubbish. It was for corporate America. And America Invents Act turned us into a first to file country, changed the ground rules. So before you show your idea to someone who might take your idea, better have some evidence that you filed for it. That's what a provisional patent does. You can do it for $150 because you all are small entities, right? I'm still a small entity. I don't employ 500 people. You can get the document on the uspto.gov website. And what you do is you fill out what your idea, my idea is blah, 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 blah. You want to be as complete as possible. You file that and you're going to get granted what's called a provisional patent. Now, these things are not examined by the patent examiner. They grant them all. All it does is it dates your idea, okay? It documents it, and it gives you a year from the date that you file your provisional patent application to go ahead and file the entire utility patent application. application, which will get examined. You may or may not get a patent out of this, but you're now protected for a year. So if you're talking to someone about your idea, they could try to run off with it, except you've preliminarily protected this, even to the point where you're allowed to use the term patent pending when you've got a provisional patent, because you do have a patent pending, so not issued yet. So learn that. That's a smart thing to do in that circumstance. Caveat, you can't take that provisional application and then expand it to cover the world. If you don't have your idea adequately described in that provisional application and you come up with a better idea as the year goes on, you can't necessarily get the date of that provisional patent application. The examiner will say, I'm sorry, you really didn't talk about this in your provisional. So that's why you want to be as thorough as possible in your provisional patent application. The other thing is the rules that apply to you. They're under a huge scrutiny in medicine, and rules are applied to us that aren't applied to other professions. So you guys probably know in the US right now, physicians cannot own hospitals. Can lawyers own law firms? Yes. Can lawyers own hospitals? Yes. Can architects own architecture firms? Yes. Physicians can't own hospitals, but there are a whole raft of other things that can get you in trouble if you're not aware. So there's something called the anti-kickback statute. I outline it in that first block. Prohibits the knowing and willful solicitation, receipt, offer of payment of any remuneration in return for or to induce the referral, arrangement, or recommendation of Medicare or Medicaid business. So don't take stuff, okay? Because Rich and I, we're subject to this in the military. So I'm very, very familiar with the anti-kickback statute. People violate this all the time. The rep says, we're going to go somewhere, I'm going to fly your wife there. Oh, I've got tickets to the concert. I'm going to go to the concert. I got tickets to the ball game. Don't do it. And maybe no one finds out, but if someone finds out, you're in trouble. Because if you're doing any business with that person that involves patient care, like you're using their implants, you've now just violated the anti-kickback statute. Yeah, that's probably the most common way people do it. What are the elements? The known willful solicitation or receipt of even an offer for payment. Any kind of remuneration, or direct, overt or covert, or in cash or in kind. And again, for surgeons, it's almost always done. It's a device or a drug, and there's some kind of exchange of value. They gave you some free samples. They bought you lunch. I mean, those are violations of the anti-kickback statute. Okay, be careful. I don't know about you guys, but for our journal club, we used to have sponsors for that, and they would pay for dinner. Oh, no. That's a violation of the anti-kickback statute. We weren't doing anything bad, but they can't pay for dinner because you might be using their products on patients. So beware of that, because if people want to get you, they can get you for that. The other one you need to know about is the False Claims Act. It's individuals or entities that knowingly file fraudulent or false claims that are payable by the Medicare program, okay, subject to both criminal and civil liability. You can be fined for $5,500 to $11,000 per claim. In other words, every single screw you went in, 11,000, 11,000, 11,000. Okay, and this act set up what are called whistleblowers. People who can file a False Claims Act against you, and they take home a cut of the pie. I think it's 20% or so of whatever the eventual settlement is, but it's a huge inducement to people to do so. So who's looking for fraud? Here's the list. A lot of people, all right, because it pays to look for fraud. And whistleblowers, key TAM relators in the jargon, include your employees, your former employees, your partners, your competitors for sure. So just know the rules. All right, don't take stuff. So how do you interact with an industry partner in a compliant fashion? I mentioned earlier in my lecture that that's an easier way to it's an easier path to innovation in my experience than the startup path, even though there are pros and cons to both. So you're working with industry. There's got to be what's called a bona fide business need. Okay, they can't just be paying you to do cases. You have to have been selected on a legitimate basis unrelated to referrals. What does that mean? So a common tactic, most of the big companies won't do this, but some of the smaller companies will. They'll say, oh, doctor, we've got this new implant. We'd like you to evaluate it in the field for us. We want feedback. And so for every case you do, we're gonna pay you, or we're gonna give you a SOC options or something. Folks, that's illegal. Okay, you can go to jail for that. No, you can get in big trouble. Don't do that. Unfortunately, people do it all the time. And they may or may not get caught. Whatever you're being paid has to meet the FMV test, the fair market value test, and you must be providing something of value. Oh, consulting, intellectual property. So I'll tell you another story. I was investigated by the Department of Justice 15 years ago. And so you know how Medicare publishes, here's what physicians are getting paid. So I've invented a lot of stuff. I get paid for royalties. I'm nothing to hide there. The numbers were big enough. They caught the attention of the DOJ, and they figured this Foley guy, he must be doing something wrong. So they show up at my office, subpoena my computers. I have to give them the hard drives to my laptop, to my home computer, to my office computer. No hammering of them, no bleach bitting of them. I'd go to jail if I did that. So they're searching through everything. There's nothing to be found, because the way my contracts are written, I had a lawyer help me with this, so I specifically stayed away from the violating these statutes. And so if I'm being paid, there is some transaction of value occurring, and typically it's a royalty for an invention that I have a patent on. In other words, there's a whole paper trail there, establishing the value and not being paid at beyond market rates. So that protected me hugely. At the end of this investigation, I get a call, and my attorney says, they found one thing. I said, really? What did they find? They said, well, they found a dinner where they think you got overpaid and violates any kickback. I said, really? I said, I'm a cheap date. I don't drink fine wines. I don't order the most expensive thing on the menu. It was at a course in Mexico, and 5 spine surgeons from different universities went out with a company, and the bill for that meal was $500. Well, it was 5,000 pesos at the time. So they found what they thought was a $5,000 dinner bill, because the company exec who had taken us out, when he won't name who it was, filed his expense report with the company. What did he do? He claimed $5,000, and he got reimbursed $5,000. So I showed this to IRS. Luckily, I had records. I said, no, that was $500 for $5. That was $100 I got. So keep good records. Just the rules. It is fine to do this stuff. You just have to do it compliantly and with disclosure. Watch out for indirect forms of remuneration, travel stuff and the like. Watch out for consulting or payments when there's no legitimate basis. Okay, be careful about that stuff. And bad actions have consequences. Here's a surgeon who had to pay DOJ $1.75 million. He was doing false consulting. In fact, the fellow at the bottom, whom you'll see on LinkedIn from time to time, just made a big settlement with DOJ. He was inducing surgeons to do this, and he's a surgeon himself. Okay. Be careful with kickbacks. Again, there's another spine surgeon. Why our spine colleagues do this, I don't know, but beware of the rules. So in conclusion, innovation and entrepreneurship absolutely help the field of neurosurgery advance. You're in the best position to do this as a practicing neurosurgeon because of your firsthand experience. Learn how to protect your ideas. Learn what the rules are governing your ideas. An easy way, or an easier way, to start this kind of activity is to work lawfully with industry. Although it's more risky and time consuming, startups can offer you greater control and greater potential reward. Thank you very much.
Kevin, come on back up. Well, a couple of things. Some of our surgeons are leaving for our careers. Oh, here you go. This, the picture, and where's the? This it?
Just legit.
Yeah. Questions? That boy, that filled your head with all kinds of ideas. Questions from the residents? How many of you are inventors? Raise your hand. All the hands should go up. Come on.
Anyone have a patent or filing for a patent? Good. Really
Yeah.
learn how to do that, gang. It's a good skill set. You know, he's right. I talked to him about this back in the Walter days.
Yeah, just
And he didn't listen.
yeah. There you go. Enough said. Sam?
Yeah, that's right, gang. Go out and change things.
Yeah, I mean, that most effective way, when I look back at Kevin's career, he's think about the stealth. How many stealth stations do we have? We have 8 in each hospital almost, and that didn't exist before. When I started, it was an idea that one of Kevin's ideas is we got to have a way of doing this without putting on a frame. Okay, there's got to be a better way of doing it. And he invented it. So unbelievable. And now we just take it for granted. Somebody actually did that.